Nervous tissue is the topic of the screencast, which you can find in Chapter 11 of your textbook. The learning objectives of the screencast are as follows. Describe the anatomy of a neuron. Contrast glial cells with the neuron. Describe each glial cell, including where it is located, that is it in the central nervous system or the peripheral nervous system, and describe its function. Lastly, contrast the structure and function of a neurolemma and a myelin sheath. The cells that are responsible for the various functions of the nervous system are called neurons. They're also referred to as nerve cells. These are not the only cells found in nervous tissue, but they are commonly what are referred to when you read the term nerve cell or you hear someone refer to nerve cells. Neurons have two very important properties. One of those is irritability. That's the ability to detect and respond to a stimulus. And the second is conductivity. After a neuron is able to detect the stimulus, it can respond and stimulate other cells by secreting a chemical messenger called a neurotransmitter. There are two major regions of the neuron that I want you to be familiar with. One is the cell body, and the second are the processes or extensions from the cell body. First, let's talk about the cell body or perikaryon. The neuron cell body, also called a perikaryon or soma. Those are all synonyms for the same thing. Basically, the metabolic center of a neuron where the nucleus and nucleolus are found, most of the organelles that you are familiar with, that's found in the cell body or perikaryon or soma of the neuron. Found also in the perikaryon or cell body are the intermediate filaments of the cytoskeleton of the neuron, which are specifically referred to as neurofibrils. These neurofibrils help maintain the overall shape of the neuron. An important organelle also found in the perikaryon is Nissl substance. And Nissl substance is a specialized rough endoplasmic reticulum which we find in neurons. Extending from the perikaryon are what are referred to as processes, shown here by the red arrows. Processes are simply nerve fibers that extend out from the cell body. These processes either conduct nerve impulses toward the cell body and are referred to as dendrites, as shown here. The second type of process or nerve fiber are called axons, and typically a cell only has, or I should say a neuron only has one axon, and an axon conducts impulses away from the cell body. And we use some specific terminology to refer to the plasma membrane and the cytoplasm of the axon. We refer to the plasma membrane of the axon as the axolemma, but it is simply the plasma membrane, just has another name. And we refer to the cytoplasm we find in the axon as the axoplasm. Axons branch at the end into structures called axon terminals, and each axon terminal is enlarged at the end into what are called synaptic knobs, as shown here in this little yellow structure that I have drawn. And within those synaptic knobs, there are these vesicles shown here in red, which contain the neurotransmitters. So again, these structures here are called axon terminals, and at the very end, that axon terminal is enlarged into what's called a synaptic knob, and that synaptic knob contains vesicles which contain neurotransmitters. Just as we can classify functionally different components of the nervous system, we can also classify these neurons functionally based on what they do. Neurons that detect stimuli and have nerve fibers that send information toward the central nervous system are referred to as sensory neurons. Neurons which are found inside of the central nervous system, in the brain, in the spinal cord, are referred to as interneurons or association neurons. And then you have neurons that are responsible for sending motor output out to effectors, and these are referred to as motor neurons. This slide here is showing the same information that the previous slide was showing, but a little more in context. What you're looking at here is a cross-section of the spinal cord and nerve fibers that transmit sensory input into the spinal cord, and nerve fibers that transmit motor output away from the spinal cord. So here is where you have your sensory neuron. Here's where you have your interneuron or association neuron within the spinal cord, and here you have your motor neuron. And now I want to add a little more detail now that you have a general idea of what this image is showing. As I mentioned, this blue color-coded neuron is a sensory or afferent neuron. Sensory and afferent neurons have dendrites that are receptors. Basically, they detect changes or stimuli. They could be, say, cutaneous receptors. This would be like dendrites in your skin, so that someone touches you, they're able to detect that increase in pressure and then send sensory input to the spinal cord, eventually going up to your brain, and your brain interprets that as an increase in pressure, and you're able to sense that someone is or something is touching you. These receptors could also be, say, visceral receptors in your stomach, and they can detect, say, changes in pH or changes in pressure deep within the body. Now let's look at the motor neurons or efferent neurons. They generate motor output that passes through motor fibers out to effectors. And effectors, again, are muscles and glands. Whenever you read or hear the term effectors, we're talking about muscles or glands. So when you decide to say move your hand, move your fingers, the muscles that are involved in that process are stimulated to contract by motor neurons in the central nervous system. And lastly, within the central nervous system, sending nerve impulses within the central nervous system are interneuron or association neurons. They typically are receiving sensory information from sensory neurons and then communicating information and activating motor neurons, but not always. Sometimes they are communicating with other interneurons or association neurons, but what's important to understand is interneurons, association neurons communicate within the central nervous system. In addition to being classified functionally, neurons can be classified structurally. And when I say structurally, I mean by the number of extensions or processes that they have. A multipolar neuron has many extensions or processes. So if we look at these two examples, right, they each have one axon, but they have multiple dendrites. So these are referred to as multipolar neurons. Going back to the image that I just showed you, notice that interneurons and motor neurons are also multipolar neurons because they have many extensions or processes. Bipolar neurons have exactly one axon and one dendrite. This example down here, here's one dendrite, one axon. One dendrite, one axon. So this is a bipolar, bi meaning two, a bipolar neuron with exactly two processes. These are rare in the human body. Typically, you only find these types of neurons in the eye or in the nose, your olfactory cells, which are responsible for detecting molecules in your nasal cavity, allowing you to have the sense of smell. Those are bipolar neurons, and you also have photoreceptors in the retina of your eye that also are bipolar neurons as well. The third structural classification of neurons are the unipolar neurons. And unipolar, uni meaning one, have exactly one process. Now you may look at this and it may look like that there's two processes, but when we talk about a process, at least when it comes to classifying neurons structurally, we're talking about an extension from the cell body. And so since this neuron has exactly one extension from the cell body, it is referred to as a unipolar neuron. So don't confuse this unipolar neuron for a bipolar neuron. It has only one process or extending from the cell body, and therefore it's a unipolar neuron. So where have you seen unipolar neurons before? That's right, all sensory neurons are unipolar neurons, having exactly one extension from the cell body. In addition to neurons being found in nervous tissue, there are many other cells that support, insulate, and protect these rather delicate and important neurons, and collectively they're referred to as neuroglial cells or simply glial cells. In fact, they outnumber neurons 50 to 1, but unlike neurons, they cannot generate or conduct nerve impulses, and they have the ability to divide. Neurons lack that ability. There are 4 neuroglial or glial cells in the central nervous system. They are the oligodendrocytes, ependymal cells, microglial cells, and astrocytes. Let's talk about the structure and function of each of these glial cells of the central nervous system. The astrocytes are these star-shaped cells. Astro means star. Here you have an astrocyte here. This is a neuron here. Notice how the astrocyte has these processes that reach out and attach to the neuron, and they also reach out and attach to nearby capillaries. So what do these astrocytes do? So first, they provide a supportive framework because they sort of anchor the neurons close to the capillaries that provide nutrition and oxygen for those neurons. They also have the ability to modify the chemical environment around the neuron. They can take up certain substances, they can release substances, they can convert glucose to lactate, for example. And most importantly, probably, they form the blood-brain barrier, which you'll learn about later on in the semester. Notice how they wrap around the capillaries, and they form a barrier between the blood in the capillaries and the interstitial fluid that bathes the neurons. And so they help regulate substances that pass between the blood and the interstitial fluid that bathes the neurons in the central nervous system. I mentioned that these astrocytes have the ability to regulate the chemical environment around the neurons. Another way they can do this by secreting growth factors that can regulate the metabolism of the neuron. And also, interesting enough, where you have damage to the central nervous system, you'll tend to find large concentrations of these astrocytes in the form of scar tissue, or what you would call a sclerosis. The second glial cell of the central nervous system is the microglia. Microglia are these spider-shaped glial cells that basically function as immune cells in the central nervous system. So the central nervous system, your brain and spinal cord, are actually sort of separated from your immune system. So your normal white blood cells, T cells, B cells, etc., actually don't have access to the central nervous system. The central nervous system has its own police force or security, shall we say, and it's in the form of these microglial cells. And these are phagocytes. If you remember what phagocytosis is, that's the ability of cells to eat solid particles like other cells. So bacteria, viruses, that get into the central nervous system, these cells can basically gobble them up. The third glial cell are the ependymal cells. So in the central nervous system, you have these cavities, and these cavities contain cerebral spinal fluid. The ependymal cells line these cavities, and they have these extensions of their cytoplasm called cilia. And that cilia beat back and forth, and they circulate that cerebral spinal fluid throughout the ventricles of the brain and also the spinal cord. And finally, we have the oligodendrocytes. I know it's a long word, just sound the word out. It's spelled as it sounds, oligodendrocytes. These cells form myelin sheaths around the nerve fibers, axons specifically, in the central nervous system. They have these processes that reach out and wrap around the axons, shown here in yellow. And so all these multiple oligodendrocytes form what's called a myelin sheath. And at this point, think of a myelin sheath as, say, the rubber around an electrical wire, where the electrical wire would be like, say, the axon, and the rubber around it that insulates it would be the myelin sheath. So those are the glial cells of the central nervous system: the astrocytes, microglial cells, ependymal cells, and oligodendrocytes. Make sure you are familiar with the functions of all four of those glial cells, and that you know they are found exclusively in the central nervous system. In the peripheral nervous system, so this is outside the brain and spinal cord, we have two glial cells: the satellite cells and the Schwann cells. So let's look at each of these cells. Satellite cells have similar functions in the peripheral nervous system as the astrocytes have in the central nervous system. They control the liquid, or chemical, I should say, environment around the neuron. They also provide some electrical insulation to that neuron, and overall control the movement of substances between the extracellular fluid and the neuron in the peripheral nervous system. In the peripheral nervous system, we have these cells called Schwann cells that, similarly to the oligodendrocytes in the central nervous system, form the myelin sheath, but they form it a little differently. Rather than having extensions that wrap around the axons of the neurons, the entire cell wraps around, right, as I'll show you in just a few moments, forming the myelin sheath. But in addition to forming the myelin sheath, they also form a structure called the neurolemma. So they form both the myelin sheath and the neurolemma in the peripheral nervous system. Now I'm going to talk a little bit more about these two structures so that you understand how they differ in terms of their structure as well as their function. Let's talk a little more in depth about the myelin sheath. So the myelin sheath is composed mainly of lipid because the myelin sheath is composed basically of the plasma membrane of the cells that form it. In the case of in the central nervous system, it's formed by those extensions of oligodendrocytes, and in the peripheral nervous system, it's formed by the Schwann cells. But primarily, it's composed of the plasma membranes of the oligodendrocytes and Schwann cells, which are composed of phospholipids. Therefore, these myelin sheaths are composed mainly of lipid. Recall that in the central nervous system, these oligodendrocytes have little extensions that wrap around several nerve fibers. So you get these tight wrappings, mainly composed of their plasma membrane, forming the myelin sheaths. In the peripheral nervous system, the entire Schwann cell wraps around the axons like a jelly roll. And notice that right up against the axon, you get these really tight wrappings. So there's very little cytoplasm here. It's mainly or almost exclusively plasma membrane. And so the remaining cytoplasm is basically forced into this outer wrapping here, where these tight wrappings, well, that's the myelin sheath. This outer wrapping, which is mainly composed of the cytoplasm of the Schwann cell, is the neurolemma. So as I mentioned, this outer wrapping is where the nucleus is going to be pushed in most of the cytoplasm. And that neurolemma basically provides like a little cushion of protection for the nerve fiber. Now, there are no neurolemmas in the central nervous system. So why do we have them in the peripheral nervous system? Well, we need this protection. It's not a lot of protection, but it's some protection in the peripheral nervous system because unlike the central nervous system, we don't have bone protecting nerve fibers in the peripheral nervous system. Your entire central nervous system is encased in bone, your skull and your vertebral column. Your peripheral nervous system nerve fibers are completely exposed. So we have this little cushion of cytoplasm, if you will, in the form of a neurolemma that provides some protection for the nerve fibers. This is just a sort of frontal section just showing the same thing. Here you have your myelin sheath providing the insulation, these tight wrappings, and then on the outer wrapping, which is where most of the cytoplasm and the nucleus is found, that's your neurolemma, both formed by the Schwann cell. So when we look at the myelin sheath that covers the length of an axon, notice that you have some areas where the nerve fiber is exposed, and those are referred to as nodes of Revere, where we have the Schwann cell, or if this was in the central nervous system, the oligodendrocytes covering the nerve fiber, we call those internodes. So this is an internode, that's an internode, that's an internode, etc. The little spaces where the fiber is exposed are called the nodes of Revere. And this is going to play a very important role in the transmission of nerve impulses or electrical signals down the axon, as we'll see later on. One other feature I want to point out, and that is the funnel-shaped axon hillock. This is basically the beginning of the axon, and it plays a very crucial role in the initiation of a nerve impulse, which is how neurons communicate. This axon hillock and this initial segment of the axon play a very crucial role in generating nerve impulses, as you'll see later. Nerve impulses are initiated in that axon hillock, and then they move through the axon. And some of the factors that affect conduction speed include the size of the axon. The larger the axon, the faster the nerve impulses are conducted. And if a myelin sheath is present, it greatly increases the transmission of action potentials. Not all axons have myelin sheaths, but those nerve fibers where speed is very important, such as the control of your skeletal muscles and sensory signals for vision and balance, those nerve fibers are myelinated. Now let's review the objectives of the screencast. Describe the anatomy of a neuron. Contrast glial cells with the neuron. Describe each glial cell, including where it is located and its function. And lastly, contrast the structure and function of a neurolemma and a myelin sheath.